I would never get on the stage with a talk like that at my institute. Karma and soul is not what we operate in science. Even epigenetics to a lot of scientists is a controversial term because it, as I will try to point out, it includes too many things, in my opinion, and there is for purposes of current understanding, there are particular focuses of it. But I will try to be a little bit vague and deliberate, just to kind of raise interests of into particular directions, and maybe there will be interesting things in it. So to a lot of people, DNA, I was investigating in outreach projects what people think about genomic predisposition and things. It's a lot of darkness associated in people's minds with genomes because you get what you get in your cards, and there's not much you can do. There are mutations, and as John elegantly pointed out, there is transposable elements, and we are unique, and we accumulate mutations as we go. But influence of that is still dark area for us, and this is an interesting thing. That twins, they are genetically identical, despite they might have mutations and accumulate them during lifetime, and they have different personalities. That is, personalities for the purposes of this talk is a little bit like soul, what I think. And but the twins, if we look at the beginning at the chromosomes, and the distribution of the epigenetic marks, which I'll say in a minute what they are, they look more similar than when they are with age. They accumulate some interesting molecular marks on the chromosome, and that is possibly associated with the accumulations of their differences in non-molecular and molecular terms. So in genetics, as John was talking about it, there are two organisms, like for example yeast and human, and they have different genomes, and genetics, that is the discipline that studies differences in these differences. What epigenetic studies is as cells from different parts of our body are dramatically different, but they have the same genome. Every cell in our body has the same genome, but cells can look drastically different. The cell from our brain, cell from our muscle, and there is a lot of different cells, and tissues, they are transformed. The way cells become different, starting from a stem cell that can go into many directions, we end up with a completely different cells that can be like cells from different organisms. That is what epigenetic studies. And the word epigenetics started in Edinburgh, in Scotland, with this elegant gentleman, Conrad Waddington, who proposed the theory of epigenetic landscape. And this is, he was also quite good at drawing, as you can see, and he was broadly interested. He was a philosopher, he was a left-wing political kind of thinker, and so on. So he proposed that a naive cell at the top of the hill travels down, and there is some hypothetic landscape that it goes through, and it accumulates certain characteristics as it goes down. So it's like it irreversibly become particular cell type, and that's is quite a broad. This became very popular now, this theory, because it with stem cells that were not known at that time, that became very good paradigm for explaining this phenomenon, how cell differentiates and accumulates characteristics and functionality. So he also was a critic of Darwin's evolution theory, and there's many interesting things. I'm quite interested in this man. Then if we look, it's also his drawing. What he implied that there is, inside the hill, there are certain mysterious molecular factors that create this landscape. So there are some molecules which he didn't know at that time that make this landscape, and they can change. This landscape also is flexible. Cells affect this landscape as they go on, and so on. So what we know now is that DNA is certainly very important and has very elegant mechanism of action. But there is also what is next to DNA. There are these proteins called histones forming nucleosomes together with the DNA. And these proteins, they wrap the DNA. There's 2 meters DNA in each tiny cell. And these proteins, they seem to be at the very core of anything that has to do with DNA. Anything that needs access to DNA. To read DNA, you need to deal with these nucleosomes. And these nucleosomes, there's 8 proteins inside each bead. These beads are uniform. All genome goes twice, around each bead. And therefore, it's compact on one hand. And also, these proteins, they have a lot of information, valuable information. I mean, all proteins in the cell have a lot of information. Because these proteins are close to the DNA, they were first implicated in having this epigenetic effects that we are talking about. So epigenetics to a lot of people is mostly what is to do with this either DNA modification. There's DNA modifications like cytosine methylation, and there are more discovered now. And there are also modifications, it's a covalent modifications, chemical modifications of the proteins. There's a particular amino acids that get acetylated. There's a particular chemical group sitting there. And that changes the property, this changes the charge of the molecule, and therefore interaction with DNA, with other proteins. And there is also a specific recognition of these little flags, little marks. So this creates an incredibly rich array of information, which is very difficult to calculate, but a lot of people do it. And it still doesn't, in the beginning, there was a theory of epigenetic code. They thought there is, apart from this very simple code with 4 letters, A, C, T, and G, in various combinations, that there is also a very complex code with these modifications. It's an elegant theory, but still, it's not understood well. A lot of people are disinterested in this theory nowadays. So this is a kind of a cartoonish model of how there is, what's the link between what we eat or what microbes produce, as Dmitry was telling us about. In my opinion, epigenetics is connecting what we eat and what we get as a result in our organism. So there is a machine producing these epigenetic modifications from food. There is folate, and it's the precursor, the chemical precursor of this modification on DNA. And therefore it is important what we eat. Not only for ourselves, but also for next generation. So importantly, this information that sits on the DNA, sits on these proteins, it has a memory. That's why epigenetics is important. And for example, it's not only humans, it's all other organisms. For example, in honeybees, this particular compartments which are supposed, any larva of the bee can become a queen bee. It depends on the diet. So the ones that will be royal, they're fed with a particular food called royal jelly, and that was shown to have inhibitor, a preventer of a particular enzyme in our body. And this is the enzyme that lays this epigenetic mark. So it has a and the queen bee looks completely different from a normal bee by size, by the function, everything is different. So this is very important. There also were studies about diet and and outcomes. The life expectancy was surprisingly, it was shown that in Sweden somewhere, there was they studied the population and put together the information about which families were poor in the generation of the grandparents, grandparents, and the grandkids. For some reason, in the poor families, their grandkids were living longer. It could be to do with bacteria. There's probably more research needed, but it's definitely what we eat affects us. And in mice, for example, because in mice we can do direct experiments, it was shown that there were genetically identical mothers that were fed different diets, and it turns out that completely changed how the mice, the children come out, and that is linked to these epigenetic modifications. It's important for ourselves what we eat, and it's important because it affects the chemical modifications in our cells, and it directs how our tissues come together, and it directs everything. Especially important to our souls, like souls for the purposes of this talk, is the personality, and we associate it with the brain function. And there are some facts, interesting facts, connecting epigenetics in this very narrow sense of the word, the epigenetic modifications, chemical modifications of DNA and proteins next to DNA. So here they were shown that memories are connected to gene expression. Gene expression is the thing that is very strongly connected to epigenetics. Then there is also mental illnesses and addiction to alcohol and drugs was shown to be also correlating. It's difficult to do experiments in humans, and it's hard to study this phenomenon in animals, but there is a strong correlation between things. The suicide rate it was also correlated, and child abuse, mental disability, different types of mental disabilities. Then there is schizophrenia, as I said, different mental illnesses. And a very elegant studies was shown that cocaine addiction, repeated use of cocaine leaves a particular chemical modification in the regions, in the big regions of the cells of the brains in the patients. And this is something that is not in your DNA, so your grandkids may not get it, but it's a quite long-lasting thing. So this is a more gentle way of regulating things than by the DNA. And this is, sorry for the quality, this is kind of the artistic representation which my friend made in Photoshop. It's a metaphor for having different DNA. We can be different and engage in different activities and affect each other, and that is broad. I mean, there should be other people as well. Let's focus on one individual. So now I will talk a little bit more close to molecular biology that I'm involved in. And I'm interested in how chromatin state is regulated. So chromatin is this bead-like structure of DNA that is wrapped around these proteins. And as I pointed out, there are chemical modifications, for example, this acetylation, and that can come and go. There are enzymes that lay this acetylation in certain places on the proteins. There can be many acetylations in one protein. Actually, all proteins in the cell are acetylated. And these acetylations, they serve as marks for enzymes that copy our DNA, that make a soft copy RNA molecule from which protein is originating, and so on. And you can see the structurally is different. You know, there are types of chromatin. There's open chromatin, it's called euchromatin, and heterochromatin is closed, and you can see they're different. You know, when they're more compact, you can see a darker staining. So yeah, this is, you can't really see much here, but this shows how complicated things are. There are a lot of metabolic reactions in the cell, and they are very interconnected. So anything, any chemicals that are in these pathways, they can be linked to other parts, and in the end, it's a very difficult, I think epigenetics will be very difficult to calculate what's going on. And a German person, long before DNA structure was proposed, Schrödinger, in a very interesting book, What is Life, he proposed that there will be new methods in physics that will allow to integrate the knowledge from biological matter in a similar matter. And the person who I think will be also very famous, who just left us, Vasily Agrysko from Paris. Originally, he is from Ukraine. He knew physics very well as biology, and he, in his literature, if you have time, please read. He tries to put quantum theory as something that will help to understand biology of the living matter in future. But going back to more simple things, so this is the chemistry of acetylation. There's acetyl group is this small group on the lysine, which is amino acid, the building block of the protein. And it was shown that in cancer, which is also, it's a fate, or in a way, it's the karma of the tissue. Something happens to the tissue that it starts to have to change the way molecular mechanisms are in the cells, in the affected cells, and it's very hard to understand what is going on. It was pointed out that acetylation in general is when there is low acetylation. When the cells lack acetylation, the prognosis is worse. It's very difficult to treat cancer like that, and the outcomes are very poor. Therefore, I was interested in what is, how the acetylation is happening in general, like without going into each particular acetylation. And so, as I pointed out, that the more acetylation there is, the more open is this bead-like DNA structure. So because of this phenomenon that the cancers are more aggressive and happen more often in their in the situation where poor acetylation, there are cancer drugs that try to prevent removal of acetylation. They're called HDAC inhibitors. It's a new generation of drugs. They also have issues, but they seem to be quite tolerant compared to other chemotherapy. So we decided to see what happens when we treat cancer cells with HDAC inhibitor, with this type of drugs. What happens with molecules, with the small molecules in the cell? We wanted to study metabolome of these cells. So we used mass spectrometry. This is quite a complex device, which basically aims to calculate how many molecules of which sort we have, and what molecules can we find. There's multiple things you can do. The devices, they get quite advanced nowadays, and it's they can do more and more. Before, people mainly looked at different proteins and their modifications. Now it's possible to use it for metabolomics. Metabolomics is the quantification of metabolites. Metabolites is small molecules in the cell that are exchanged between food, bacteria, and our own cells. And they, as I will explain a little bit later, they are also they are carriers of information for the outcome, for the karma of what is going to happen with cells and tissues. So what we find using this analysis is that in the cells that were treated with this cancer drug, HDAC inhibitor, we start to lose a lot of acetylated small molecules. These are amino acids which can be used, some of them can be used as building blocks in the proteins in the cell. And for some reason, you lose the acetylated amino acids. And we thought, this is interesting. I mean, nobody ever found any role for this acetylated molecules. Maybe they're the carriers of this acetyl group. Because if we treat the cells, if we prevent removal of acetylation, if we keep acetylation high in the cell, these probably are the molecules from which this acetyl group is coming. So we use another big machine for these purposes. It's called reverse phase proteinary. This is a lot more simple than mass spectrometer, but it allows us to see how many acetylations of different kinds we have in the cells. And if we look at the result, it looks like in our cells, this red bar is how much acetylations we have in the cell when we treat cells with a drug. And as we expect, we have a lot more acetylation, because that's what these drugs do. They prevent removal of acetyl group. But interestingly, when we take these acetylated amino acids and feed cells with them, we have an increase. We are able to increase the acetylation in the cell even more. And that, for me, it means that potentially it could be a useful therapeutic agent for cancer cells, because they are non-toxic. I mean, acetylcarnitine is used by sportsmen, as vitamin pills, and other acetylated amino acids, they're not toxic. So that is, I think, is an interesting finding, despite we still don't understand exactly how they work. And then a little bit about what I am recently got interested, because I like this holistic ways of looking at things. I think that because the food is so important for controlling our fate, it might be that as Dmitry alluded to, that possibly at some points in our history we were eating different things. When we were more close to nature, we were picking herbs, mushrooms, and other things. Our microbiomes was different. And nowadays, because we thought, okay, the species like wheat, the meat from cows, these are the good things, and we only produce this type of food, and the majority of the population only eat these things. Maybe there is something missing from the diets which we did not acknowledge. So it's my theory is that the fact that we have a lot of problems like cancer, we have other things, it could be to do with the fact that we don't use a lot of green things that live around, the mushrooms and so on. So a new project that we started, we tried to collect herbs that were used in medicine traditionally, like in old-fashioned societies or by shamans in different cultures. For example, so the idea here is that we take cancer cells, we take different cancer cell lines, and we treat them with extracts from different plants or fungi, and we see if there is a selective effect of this particular herb. Because animals, I mean, if you ever looked at cats when they're not well, they walk and they eat a little bit of this herb, a little bit of that. They smell, they trust their smell, they trust their taste, and nowadays we cannot do that anymore. We only trust information. So it looks like in our hands we took different kinds of plants from different countries, and we already see that there are cancers that respond very well to particular herbs. These are 3 Mauritian herbs from Mauritius that we've collected them there, and we are now trying to purify to see what compounds can we purify from the cells when we do purification, which sets of molecules are killing the cancer cells. It's important, of course, that they don't kill other cells, because a lot of herbs, they have toxic compounds, and it's important to maybe purify the toxic from non-toxic, or maybe keep the mixture complex and try to see maybe you need a lot of molecules, because the pathways are so complex.
Can I ask a question?
Sure.
You say that these plants have a killing effect on cancer cells. That's based on an in vitro assay.
In vitro assays, yeah, we are not allowed to treat ourselves. Animals, yes.
You're not allowed to inject these things in animals. And if you inject, what you do?
We did not do these experiments. There's a lot, yeah, there's ways we can do. I think especially interesting are the herbs that are admittedly non-toxic, because there are a lot of herbs that are, that we know that they're not toxic. So we could use them as food supplements in people. And maybe it's not therapeutic, but especially with a cancer like esophagus, the cancer of esophagus, where you have all repertoire of molecules going inside the tissue, I think that may have a lot of promise, these things. Because animals experience, sometimes they are reproduced in humans, sometimes not as well. Also, I think for the sake of time, I will stop here. I will thank people that were doing the experiments, and all the people who contributed their reagents and things like that. And thank you very much for your attention. And I would like, yes, sorry, I also would like to collaborate with everyone on.